Hello children, let me continue with yesterday's lesson. Today I will show you what happens when a cell is immersed in an isotonic, hypotonic and hypertonic solution. Now the solutions outside, they are salt solutions, okay, in different concentration in comparison to the cell, in relation to the cell. So here the salt concentration is more or less same as the fluids inside. So you can say the cell, this is a plant cell dipped in isotonic solution. What happens is there will be no change as the molecules of water leaving and entering the cell will more or less be the same. So neither the cell shape, size, nothing changes. Then what happens is, the second one is the plant cell is dipped in a solution which is hypotonic. Yes, the solution outside is hypotonic. Hypotonic, lower in concentration in relation to the cell. Lower in concentration, meaning it has more water, no? More water outside. So what happens? Water starts entering the cell. When it starts entering the cell swells up and it becomes turgid. This process of water entering the cell, this is known as end osmosis. Then the third type, what you have is the cell is immersed in a solution which is hypertonic. The concentration is much higher, okay, in relation to the cell. That means the concentration inside the cell is less. No? So outside the solution is hypertonic. So in this condition, what happens is water leaves the cell. When water leaves the cell, it shrivels up. Yes, because water leaving the cell, that means what is happening? Exosmosis. And when the cell shrivels up, it becomes loose, and that condition is known as flaccid condition or flaccidity. Okay, the cell becomes flaccid, loose. Okay, so these are the three conditions when it is dipped in three different types of solutions. Okay, this was the next thing that we did, active transport. Now what is the active transport? What is it? Passage of substances. But passage of substances from its lower concentration, okay, to a higher concentration. That means when it has to move from its lower concentration to a higher concentration through a living membrane. And here, when it has to move from a lower concentration to a higher concentration, energy is required, and energy is given by the cell. Now, what usually moves? The nutrients such as nitrates, sulfates, potassium, zinc, and manganese. These are the nutrients that usually move from a lower to a higher concentration. Okay, now they cannot pass through the cells of the cell membrane of the roots easily, because the roots already have a higher concentration of these inside the cell. As you know, the cell sap concentration is higher than the surrounding water because the root maintains a higher concentration inside the root because it has to absorb water. As so long as this is maintained a higher concentration inside the cell sap of the root, then water is absorbed. So as the concentration is already higher inside the root cells, so these ions have to be forcibly carried inside if they have to move from a lower to a higher concentration. Okay, because I said inside it's already high. So how they are taken in forcibly? This is by applying energy, that is ATP. Now, next type of transport we did was passive transport. I told you it is nothing different than diffusion. It is same. It is called passive because no input of energy is required and the molecules move from higher concentration to a lower concentration. No energy is required. That's why it is called passive. And the other one is called active transport because it requires energy. Turgidity and flaccidity. When root hairs are surrounded by soil water, see, the root is surrounded by soil water, osmosis occurs, and that's how water enters the cell sap. Inside the root it enters and it becomes fully tight, and that condition is known as turgid. Okay, condition turgidity. Turgid condition is known as turgidity when it is when osmosis has taken place and water has entered the cell. Now, a turgid cell is stretched under pressure from inside. That means it is very tight from inside. Lots of water. So from inside only it is very, very tight. It presses against the cell contents towards the center of the cell. That means it also presses against the cell contents towards the center of the cell, contents of the cell. That means what presses against the cell contents? It is the cell wall over here that presses against the inside of the cell, the contents of the cell. Okay, but at the same time, what happens is when cell wall is applying pressure inside, the contents of the cell, they also apply an equal pressure outside. Okay, now the pressure exerted by the cell contents from inside, what it contains, the cell contents, when it applies a pressure outside on the cell wall towards the cell wall, that pressure is called Turgor pressure. When the wall applies pressure towards the cell contents inside, that is called wall pressure. Now, if the pressure is balanced, the cell pressure, okay, the wall pressure applied by the cell wall and the turgor pressure applied by the cell contents, when it is balanced, then the cell remains nice and turgid, okay, if it is balanced. But if the turgor pressure, that means the cell contents from inside, if their pressure is higher than that of the wall pressure, then the cell will burst. Okay, it will burst. Now, water moves into the cell due to osmotic concentration of the contents. Due to osmosis, water moves into the cell. So this is the pressure. Now, you know what is wall pressure and turgor pressure. Okay, wall pressure is the pressure applied by the cell wall on the cell contents inside. That is called wall pressure. Turgor pressure is the pressure applied by the cell contents on the cell wall. That is called turgor pressure. So long as it is in equilibrium, say as long as it is not more, as long as the turgor pressure is not more than the wall pressure, then it remains in a turgid tight condition. But when the turgor pressure becomes more, what happens? The cell wall will burst. Okay. Now this is a cell, a plant cell in fresh water. When it is in fresh water, you can see that this is the cell wall, then the plasma membrane. Okay, inside is the vacuole. Okay, with the green line showing the vacuolar membrane. Okay, the nucleus. When it is in fresh water, the cell is nicely stretched. The cell wall, the plasma membrane is next to the cell wall in a distended tight condition because it is in fresh water. Now, the same cell, when it is put in a hypertonic solution, say you have put it in some amount of salt solution, that means that solution is higher than that of the cell sap, the cell fluids inside. What happens? Slowly, the cell wall from the plasma membrane will leave the cell wall. Can you see the black one leaving the cell wall? It is slowly leaving the cell wall because this is a hypertonic solution. So what is happening? Exosmosis, no? Water is leaving. So the plasma membrane leaves the cell wall. The cytoplasm also starts shrinking, okay? It starts shrinking slowly, then it has shrunk more over here, okay, the plasma membrane, because exosmosis is taking place. And finally, this is the state of the cell where so much of exosmosis has taken place that this has actually become flaccid. And see, the plasma membrane is here, and this is the vacuole. That means the water has left the cell fully. And this condition of the cell is known as the plasmolysed cell. Okay, when water has come out from the cell and the plasma membrane it is away, it has contracted, okay, it has left the cell wall. This is called, okay, the plasmolysed condition of the cell, when the plasma membrane has left the cell wall, yes, because of the exosmosis, water has come out of the cell, yes, and this is a plasmolysed cell. There's lysis, that means it has left the cell wall, the plasma membrane, yes. Now, this plasmolysed cell, if it is kept in, again, fresh water or a hypotonic solution, then what happens? It will come back to its original form. When it comes back to its original form, it is called the deplasmolysed state. When the plasma membrane again comes close to the cell wall, and again the vacuole is fully, full of water. By absorbing water, it comes back to its original form. This is called the deplasmolysed state. Thank you. So here what you actually saw was that the plasma membrane has withdrawn from the cell wall. This is the cell wall, plasma membrane has withdrawn, okay, and that time we call it the plasmolysed state. But again, the reversal of plasmolysed cell or plasmolysis, reversal of plasmolysis. This is a plasmolysed cell, it's called plasmolysis. But reversal of plasmolysis is when we put it in again a solution, or say again back to say fresh water or hypotonic solution. Then again endosmosis will take place. Yes, water will enter the cell and the cell will get deplasmolysed. It will come back to its original form. The cell membrane again has gotten back next to the cell wall, and it is in a very tight distended condition. This is known as deplasmolysis. Okay, now the terms, this is how they are defined. Turgidity is the state of a cell in which the cell wall is rigid and stretched. In which the cell wall is rigid and stretched by an increase in the volume of vacuoles due to absorption of water. Then it is said to be turgid. So how does it become turgid? By the increase in volume of the vacuoles due to absorption of water. Now the next term is plasmolysis. It is contraction of the cytoplasm from the cell wall. It is contraction of the cytoplasm from the cell wall caused due to withdrawal of water when placed in a strong hypertonic solution. So that has happened. Now flaccidity is the condition in which the cell content is shrunken and the cell is no more tight, no more turgid. Then the cell is said to be in a flaccid condition, when the cell is no longer in a flaccid condition. So these are the terms: turgidity, plasmolysis, and flaccidity. Thank you.